going to talk about is lipids. The lipids is a type of organic molecule that is also an energy storage molecule, just like starch or sugars, I should have said, not just starch. But it's harder to break down, and therefore it's for longer term storage. These guys are also hydrophobic. It means that it does not dissolve in water, and we'll talk about why that is in just a bit. Okay, so if you look at the basic structure, so the basic structure has a glycerol head and fatty acid chains that are off the end, so it looks kind of like that. This is a more, a better drawn out version of that. That is, this is the glycerol head. Notice glycerol is somewhat like a sugar in the sense that it has hydroxyls, but it doesn't have that carbonyl group, right? So glycerol by itself could, somewhat disassociate and maybe slightly mixed with water. However, what happens is that hydrogen comes off of there, as well as this hydroxyl over here, via dehydration synthesis, joining the fatty acid chains to the glycerol head. So if that happened 3 times, you would form these, what they call ester links, between the 3 fatty acid chains, and this would be called a triglyceride. This is a monoglyceride. All right, so you could have a diglyceride as well. All right, so dehydration synthesis makes these molecules, but these molecules are representing one lipid. Now, they do not bond to each other. In other words, another lipid would not come along and bond to this lipid, or this one would not bond to that one. So lipids do not have that monomer polymer system that we were just talking about with the carbohydrates. So that is a little bit of a difference. Now the other thing is because this part, once it's joined, the glycerol head, once it is joined to the fatty acid chains, notice there is nothing that can disassociate, like the hydrogens from here could disassociate and form with water. And there's nothing over here that could do that either. So there's nothing that could be charged or disassociate and mix with water. So these guys do not mix with water at all because they have no charged portions to their structure. Now in this case, the fatty acid chains are completely filled with hydrogen. That means that every available covalent space for every carbon is either connected to another carbon next door with a single covalent bond and with the hydrogens on the other single covalent bonds. Or at the end, it would make a carbon with 3 hydrogen bonds. This is called a methyl group. The rest of them are completely full. This is what we call a saturated fat. And saturated fats are solid at room temperature, and it just means that all the carbon's valence locations are filled with hydrogen. Okay, so they would all be filled with their hydrogen or connected to another carbon. Okay, now what can also happen though is sometimes a hydrogen may be missing. In other words, I could take off some of these hydrogens, and if I did that, the covalent space then would have to make a double bond between the carbons. And when that happens, it makes the molecule actually bend, or that is, these tails will bend, and because of that, they do not lay down on each other flat. So in the saturated ones, these hydrocarbon tails are relatively flat. One saturated one would lay just on top of another one, just like stacking a bunch of paper, which is why it's a solid at room temperature. But if these tails are not flat, they don't lay flat on top of each other, and therefore this substance is a liquid. All right, so unsaturated fats are a liquid. So those are the main 2 classes. Now there's 2 other classes that are very important. One is phospholipids. Now phospholipids, oh wait, this is showing you the saturated versus unsaturated. By the way, if I were to make a fat, so if I took a saturated fat like butter and I were to unsaturate it, that is, remove or polyunsaturated fat, that means that I remove those fats. If I were to do that or dehydrogenate it, it would make it a liquid, but it's still, you know, basically started off as a saturated fat. So just so you know, that's how they make those like whipped butters and things like that. They remove the hydrogens as opposed, and it still started off as a saturated fat. So oils, just by the way, liquids or oils are easier to break down because of these, bends in the saturated fat tails or the fatty acid tails. Makes it easier for enzymes and water, etc., to get in and help break down these fats. So they're a little easier to break down in your body. All right, so the third class, as I started to talk about, and now I'm returning, is the phospholipids. All right, phospholipids. And what we are going to talk about in this case is the phospholipids are built the same way. In other words, they have the glycerol head and they have the fatty acid tails. They could be saturated or unsaturated, and in this case it's showing you one of each. But it also has a phosphate group, and then it has a little cap called choline. Now this phosphate group, you might recall, has some charges to it. So notice it has a charge. That means what? It means that it can dissolve in water. So what I've done what happens with these is that it creates a polar molecule in regards to solubility. In other words, the head of the phospholipid dissolves in water, and the tails, or the fatty acid tails on this part, do not. So what would happen if I mix it with water? So if I mix it with water, what's going to happen is water will dissolve the heads and the tails will face each other, and it would create a void spot in the middle. Or if I keep mixing it with water, I can make little sacks of water on one side and water on the inside with a double phospholipid layer. In other words, I have the heads facing towards water on the inside and heads facing towards water on the outside, with the tails facing each other. Now this is the basis for cell membranes. All cell membranes have at least mostly phospholipids as their a double biphospholipid layer as their main matrix for the cell membrane. You can also say that phospholipid bilayer. It's correct both ways. Okay, so these phospholipids are important in cell membranes, and it is all because of that phosphate group allowing that phosphate head to dissolve, the tails to be a water exclusion area, and that means that you can then have a cell, basically. All right, and lastly, the last group is called a steroid. Now, steroids don't look anything like the phospholipids that are the lipids that we've been talking about. They do not have a glycerol head, and they do not have fatty acid tails. They are a ring structure like these, and you notice estradiol and testosterone are examples of steroids. The reason why they're put in this class is because they are not soluble in water. And in biology, or where you would find it in a biological system, is going to be mixed with the fatty acids of the phospholipid bilayer of cells. So estradiol, testosterone, etc., prostaglandin is another example. Those things will dissolve or hang out basically with other hydrophobic substances, and in biology mainly that's the fatty acid tails of the phospholipid bilayer of the cell membrane. So that's where you're going to see these, and you can see that they don't look anything like what we've been talking about.